First, we'd like to introduce the observation results of the non-diapausing pupa of Papilio bianor. Various pupae of the subgenus Ochilides including Papilio parus have characteristics that can be visually distinguished from the pupae of other subgenus Papilio species with some exceptions. Furthermore, among these species that inhabit the temperate zone, there are species that form diapause pupae. In the case of Papilio bianor, the color of the non-diapause pupa is almost uniform throughout the body, whereas the diapause pupa is characterized by whitening around the line on the body side. And it is easy to distinguish diapause pupa from non-diapause pupa. Now, we will explain how to install the butterfly pupa in the coil. First, the butterfly pupa is fixed inside the styrene sample tube, and this is attached to the auxiliary tool for fixing the sample in the coil. Next, attach the auxiliary tool with the butterfly pupa so that the butterfly pupa is in the center of the coil. Then, attach this coil to the imager equipment and we are ready to take an image. Furthermore, various adjustments and imaging conditions were set for the equipment from the computer, and imaging was started as soon as the preparations were completed. This time, preliminary imaging is performed under some conditions. Images with the imaging position changed every 0.25 mm are repeatedly imaged multiple times. And these are superimposed to create a fine textured image. Next, we will explain how to capture these data. In the proton explained earlier, as the energy of electromagnetic waves given from the outside, that is, the amplitude or irradiation time, increases. The number of protons those are opposite to the static magnetic field increases, and at the same time, the direction of magnetization of the entire protons will fall in the opposite direction of original direction. Here, if the irradiation of electromagnetic waves is stopped when the direction of magnetization is tilted by just 90 degrees, the proton will return to the previous state while releasing energy in the form of electromagnetic waves. Here, the time it takes for the magnetization strength, which has the same direction as the static magnetic field, to recover to 63.2% of the original strength is called the spin lattice relaxation time. In order for an atomic nucleus once excited by external energy to return to its original state, it is necessary to release its energy to surrounding molecules and atoms. This energy is emitted in the form of electromagnetic waves, and the total energy of the atomic nuclei is attenuated. The time required for this transfer is such that in a molecule such as fat, which has a large molecule shape and hydrogen atoms are difficult to move, energy is quickly transferred to surrounding atomic molecules, and the time is shortened, resulting in a strong signal occurs. On the contrary, in the part containing many small molecules such as water molecules, the movement of the molecules is intense. So the transfer of energy from one molecule to another does not work well, and the time until the energy returns to a low state. The longer the signal, the weaker the signal from those parts. For the above reasons, the T1 weighted images that created an image based on the difference in spin lattice relaxation time. The signal of the part containing many large molecules becomes stronger, so the picture shows macromolecules such as lipids whiter. On the other hand, at the moment when the magnetization collapses by 90 degrees, the magnetization rotates as one as a whole at first. However, it gradually splits into multiple magnetizations with different rotation speeds, and the overall magnetic force becomes weaker than when rotating with one magnet. The time it takes for the strength of this magnetization to reach 36.8% of the strength when rotating with the first one is called the spin-spin relaxation time. Spin-spin relaxation is different from spin lattice relaxation caused by the transfer of energy, because the magnetic environment of one proton in the tissue differs in the strength of the magnetic field applied to each nucleus due to the influence of other proton around it. It occurs when individual nuclei rotate at slightly different speeds and the magnetization becomes disjointed. When the molecule is small and the movement is large like a water molecule, the difference in the strength of the magnetic field is averaged over time. So it is difficult to make a difference in the speed of rotation, and the signal does not weaken easily. Therefore, the signal from the proton of the water molecule is relatively strong. On the other hand, in macromolecules such as fat, a certain proton is greatly affected by the surrounding protons. And the difference in the strength of the magnetic field applied to each proton becomes large, so the spin-spin relaxation time becomes short. Therefore, in T2 weighted image, which creates an image based on the difference in spin-spin relaxation time, the part with a lot of water appears whiter. As described above, various images can be obtained by combining the image of the T1 weighted image in which the high molecular weight parts such as lipids, melanin, and proteins appear white, and the T2 weighted image of the spin-spin relaxation enhanced imaging method in which the water-rich part appears white. We can get information in the living body. The data obtained in this way was saved in the DICOM format, which is a standard format for recording 3D images for medical use. From this data, it is possible to make continuous sections from various directions, and even to reconstruct a 3D image. Common methods include the 3-dimension volume rendering method and the maximum intensity projection method. The former is a method of integrating the density of objects in front of the line of sight and generating a translucent projection image, so that the appearance of the subject can be seen. The latter is created by selecting and superimposing only the part with the maximum signal strength in each of the captured cross-sectional images, and the inside of the subject can be seen through. In this video, we decided to create a sagittal section at the midline from the information obtained and show the changes over time inside the pupa. First of all, we will show you the changes in the appearance of the pupae. Next, we will show you the NMR images corresponding to upper pictures. Anatomical studies to date have revealed that the adult structure of holometabolous insects, including butterflies, is almost complete in the prepupa stage. However, the activity of the butterfly larvae had not yet subsided during this period, and it was thought that the changes could not be tracked well with proton magnetic resonance imaging. Therefore, this time, we decided to take the first image as early as possible after the larvae completed threading, and then take images every day as much as possible to track the changes in the internal structure after that. The daily images are shown with a T1 weighted image in the upper left and a T2 weighted image in the lower right. In the lower right of each figure, the number of days elapsed with the day of pupation as 0 is shown. First, we will show you how the pupae that emerged from male butterflies change over time. First, it can be seen that the internal structure also changes dramatically during molting from the prepupa to the pupa. In particular, the alimentary tract, which occupied most of the interior during the larval period, becomes smaller rapidly. Then, as the days go by, there are what appears to be cavities under the antennae and under the mouth, which grow larger and larger. It feels strange that this part becomes hollow. However, one of Inu's acquaintances, Horikawa Kori, published a photo of a pupa of Papiliozoethus with a left wing that had failed to deploy, showing that the pupa's wing was actually hollow. The next major change is the appearance of a distinct circular structure on the dorsal side of the pupa's abdomen. This structure was judged to be the testis, which is a male reproductive organ, based on the fact that it cannot be seen in the pupae of female butterflies, which will be introduced next, and the internal structure. The reason why this structure is not seen in the images immediately after becoming a prepupa or pupa is that the testis of the butterfly is divided into the left and right sides of the body during the larval stage, but after pupation, it moves closer to the median plane. It is thought that it will be possible to confirm it in the figure of the median line because it is integrated. After that, the major changes subsided, complex structures gradually began to appear in the lower abdomen, and wrinkle-like structures appeared in the chest a few days before emergence, and from the water the day before emergence. Since the image of the T2 weight image that can record the signal of is very poor, it can be seen that the water in the body has moved somewhere at this point. Next, we would like to introduce the changes of the pupae from which female butterflies emerged. The reason may be that the ovaries of female butterflies are spread out on both sides of the median plane, so even if the changes in the median plane were tracked, the changes could not be imaged. Other changes were almost the same as those of the male butterfly, and it was also confirmed that the T2 weighted image became very dark on the day before emergence. However, while the thoracic cavity expanded rapidly in male butterflies, the cavity did not expand much in female butterflies even just before emergence. This was also confirmed in other individuals photographed at the same time, and was thought to be a characteristic of female butterflies. Next, we will explain the changes in the papilio-bianor diapause pupa. Since article of Shimada in 1976, the results of investigating the conditions under which larvae become diapause pupae have been reported in several papilio genus butterflies. However, in papilio-bianor near Tokyo, such a study was not reported, so we first conducted an experiment to investigate this point. Experiments using other species have confirmed that the factors that cause diapause pupae to appear are day length and temperature, but this time, when the temperature was fixed at 25 degrees, the day length was less than several hours. We investigated whether pupae appeared. As a result, it was found that the papilio-bianor larvae around Tokyo began to appear diapause pupae when the bright time of 24 hours was 15 hours or less. And all the individuals became diapause pupae when the bright time was 12.5 hours or less. Therefore, in order to make diapause pupae reliably in summer, it was found that the pupae should be bred with a bright time of 12.5 hours or less. In addition, Kaito uses Atrophane biasa alcinois to raise individuals in various places with fixed temperature and day length. And it is strong between the latitude of each individual's habitat and the critical day length when half of the individuals become wintering pupae. We have shown that there is a correlation. And Shimada of the Institute of Low Temperature Science, Hokkaido University used papilio macae on pupae obtained by breeding in a short day. And if the pupae are stored at 5 degrees for a short period of time, emergence will vary when returned to room temperature of 20 degrees. On the other hand, if the storage period is 180 days or more, it is shown that emergence will be gathered. Next, among the diapause pupae of the same papilio butterflies, in Papilio xuthus and Papilio machii, if the storage period at 4 degrees is 30 days or less, the emergence will vary by 80 days or more when the room temperature is returned to 25 degrees. A 1974 paper by Achinos showed that if the storage period at 4 degrees is 120 days, emergence will occur collectively within 20 days when the temperature is returned to room temperature at 25 degrees. Furthermore, Achinos and Iwasaki conducted a similar experiment with Papilio protenor, and showed that Papilio protenor from Tokyo also emerged when it was allowed to spend 90 days or more at room temperature of 5 degrees. Regarding Papilio xuthus, Hidaka and Hirae, and Endo and Murakami also published research results on photoperiodism and dormancy induction using this species. In addition, Yoshio and Ishii published a paper comparing the number of days until emergence of Papilio memnon populations living in different latitudes. Hashimoto also showed that when Papilio bianor from Chiba Prefecture was used and the diapause pupae were allowed to spend 140 days at room temperature of 4 degrees, all the individuals emerged almost at the same time. In addition, Oliver conducted an experiment using Papilio polyzenes from the northern part of the United States, and when the pupae were placed at 21 degrees without cooling. If both the larval time and the pupal time were managed in a short day state, the emergence time would be summarized. Therefore, this time, for diapause pupae, a cooling period of 3 months or more was set to adjust the emergence time. And images were taken while adjusting with other species and other users of the equipment. The layout of the figure is the same as for non-diapause pupae. The blue numbers indicate the period during which the pupae were cooled. First of all, we will show you how the pupae that emerged from male butterflies change over time. First, the changes after pupation are similar to those of non-diapause pupae, but in non-diapause pupae, the enlargement of the antennae and cavities under the mouth and the formation of testicles appear to be synchronized, whereas in diapause